Okay, let's start with a sound check. Can you guys in the back hear me? Great. Can you hear me back there? Excellent. There should be handouts for this lecture, and with luck, Angie will have tracked down what happened to them. While we give her a moment to get in here with the handouts, I want to share with you something that's on Icon. I hope most of you... Huh. Well, goodness. Yeah, or she's in Madeline Shea's lab. Madeline Shea. Oh, Madeline Shea. Right, yeah. I hope those of you who are interested have found the links part of Icon so far, and it's up there in that toolbar and gives you a bunch of links. This is where you can access the Google version of lectures. This is the direct link to it. You can also search for them on Google with just 99 by saying 99-110. Did you have a question? Oh, sure. Thank you. So the links is in the toolbar about here. I guess that's dark enough now, is it? Yeah. The links is there. In addition to those, I just want to draw your attention to the fact that there are several online textbooks. If you find yourself confused about something, sometimes it's good to cross-check. And I wanted to show you a wonderful video of the inner life of a cell, but since it doesn't have Flash 8 on here, I can't. I hardly recommend to you to go and look at this video at your leisure on your own computer, since I can't do it for you here. Are there any other questions about Icon that I can give a guided tour before we get into the overview of metabolism? Okay. I will close this up then. I don't know what has happened to the handouts. I know that the secretary's had those files for quite a while, so it ought to have been here. No human activity is perfect. Come on. Some of them are less perfect than others. Come on. Open. Why aren't we opening? All right. This is being unusually recalcitrant today. Let's see. This is here. All right. All right, let's try it another way. Let's see. That's PowerPoint. Open PowerPoint directly. Sorry for the unusually high incidence of non-useful things today. All right, after much travail, and pain. Come on. I wonder if I actually need to tell it to actually quit out of that. Well, we'll see. All right. As I said, I do hope that there will be a paper version that's coming shortly. I can see that there's also been a slight change in font here in front of me, but it doesn't actually interfere with what we're trying to say. Today is one of the rare one day for a chapter lectures. It's a fairly short chapter. The overview of metabolism chapter is meant to be a roadmap at the high level. So if you're going to drive from here to San Diego, you would be very smart to start by looking at a map of the United States to figure out overall the general route there. You know, you're going to pass through Oklahoma, but you're not going to pass through Indiana, that kind of thing. And so the lecture today is that high-level view of metabolism, in which I'm going to hope to introduce you to some of the key elements that you're going to be seeing again and again and again and again in metabolism as we go through the rest of metabolism. Some of this will almost certainly be refreshers for you from previous biology courses, but everything I say today should be very firm in your minds before we start on metabolism in detail. Metabolism can be broken down into two broad parts, catabolism and anabolism. And catabolism is the process of, let's go back. Got to fix my arrow. Here we go. All right. Catabolism is, broadly speaking, breaking stuff down, breaking it down to get energy out of it, breaking it down to get parts out of it. Anabolism is the other direction of building stuff up. It's going to cost energy. You need some building blocks to do it. It's easy to remember which one is which because anabolic steroids help build up muscles, right? At least they are supposed to. And this is the general overview of that process, that you start with some kind of macromolecules, and we're going to be going through all the different kinds one chapter at a time. Breaking it down, which liberates free energy. Really? Oh, well, the system has broken down. I'm sorry. There should be a version of this available off the web very soon after the class is over. Is there anybody who's missing paper and would like some sheets of paper from a fellow classmate? This hasn't happened in my time teaching at Iowa, so I apologize for not being here. Okay, well, we'll roll ahead then. And then that free energy released in breaking things down is then captured to rebuild molecules. Broadly speaking, there are several ways you can make a living, and these things are called trophic strategies. Trophic strategies mean, what do you eat? Where do you get your energy from? The most autonomous, independent ones of all are a group of organisms called chemoautotrophs who use chemical energy. They can eat rocks, for instance. They can eat hydrogen sulfide. The second, much larger group would be the photoautotrophs, and these are everybody who can capture light energy, photo, as you can imagine. So plants, algae, all of these guys capture their energy from the sun, and then they're building blocks from the environment to make big stuff. And then the rest of us are all dependent on them. We're heterotrophs. We eat other things. And so we and every animal are going to be dependent on energy captured somewhere along the way, usually by photoautotrophs. Chemoautotrophs do not make up a useful part of the agricultural market. In general, the sources of energy that we're going to be taking advantage of are stored as polymers, and because they're polymers, they need to be broken down before they're useful. And the typical thing that's going to happen is breaking bonds with water. And this is a very small H2O down here. Because we use water to break something, it's called hydrolytic, breaking with water. Lytic, lysis is always a breaking. Hydro is our friend water. And so the enzyme amylase can take a starch molecule and break between the glucoses to liberate individual glucose molecules. Another polymer that gets used as a precursor would be proteins. And in the stomach, for instance, you have polymers of polypeptides. And the peptide bonds there need to be broken, again, by breaking it with water, liberating individual parts of the peptide until you get down to amino acids. We saw with chymotrypsin, it was a hydrolytic enzyme. And then the third, rather small polymer would be the triacylglycerides, where you have 3 fatty acyl chains on a carbon backbone. And again, a hydrolytic reaction by lipase, liberating the fatty acids to be used for the next step. That, broadly speaking, happens in the guts. Some in the stomach, some in the small intestine. It's not going to do you any good in there. It has to get into us, and that's the job of the intestinal cells that line the GI tract. There are active transporters where they invest ATP to get things in. Some of the glucose is taken up that way. There are also active transporters to take up small peptides. Usually proteins aren't broken clear down to amino acids. They're broken down to smallish things. A fairly large number of passive transporters as well that just depend on the gradient. and some things just diffuse in. Some lipids are able to just sort of float in and become part of the party. All of these things that get taken up from the gut have as their first spot that they pass through is the liver. So the GI tract has a huge amount of blood supply, and that goes through the liver first. So the liver has a first crack at it, and it gets modified and modulated there so that the best stuff gets to the right places after that. In some cases, we want to store some of these things, and we've seen some of these already and some not. If we want to store energy, there's two ways of storing that. We've looked at the structure already, the triacylglycerols. Those are the glycerols with 3 fatty acyl tails. This would be a fat cell here, an adipocyte. We can store sugars as glycogen, and this is a schematic diagram of the branched molecule of glycogen. One of the advantages of storing is fat is it's anhydrous, and so you get a lot more energy per pound. If you look at a migrating bird, it stores its energy as fat, not as glycogen. Proteins are only in a limited sense, storage molecules. If either glycogen or fat is available, those will be used by preference by an organism. But if there is stress, if an organism is fasting, if there's a fever, all of those things may lead an organism to break proteins down to be used actually for fuels and for energy. It's not desirable. That's usually a sign that things are not in the best possible circumstances. Some of the other things that we can absorb out of the food supply are the things we need to build nucleotides. We don't, wonderful, excellent. I knew it wouldn't work. He was somewhere. I'll keep talking, but they'll work hard at getting him out fast. So the precursors to nucleotides, the bits and pieces need to be in the diet, not the nucleotides themselves. Nucleotides are too essential for them to be a dietary component. So organisms make their own nucleotides, but they make them from things like carbohydrates for the sugar part, and from amino acids to provide some of that nitrogen, because if you think about nucleotides, the purine and pyrimidine rings all have nitrogen in them. So those are going to be the precursors there. And I may actually wait to get to this page because this is a weird word that you might want to get to. Right, nucleotides are too essential. This is on slide 9.5. Nucleotides are too essential to be, at the risk of whether it's in the diet or not. So every organism can make nucleotides. We have ways of scavenging nucleotides. So if there are nucleotides in the diet, we can scavenge them, but we do not depend on them. Dietary nucleotides are partially responsible for gout, for instance. If you eat too many mushrooms, you may end up with too many nucleotides in your diet and get gout. Not that that's a big risk at your age. So then if we look at how we can get stuff out of the polymers, one of the major ones is glycogen. And glycogen is properly speaking the storage form that our cells use. Starch is a storage form for plants, but we don't use starch. We use glycogen. There are some chemical similarities, but the specific form we use is glycogen. And we don't use hydrolysis to break it down. We use a process called phosphorylasis. And phosphorylasis is taking an inorganic phosphate, see that's just an inorganic phosphate ion, and using the inorganic phosphate to split one of the bonds between the glucoses. And when you break the bond by adding a phosphate across it instead of adding water, if you add a phosphate to break the bond, what comes off is a phosphorylated sugar. And so phosphorylasis will produce glucose 1-phosphate as an intermediate that can now be used for something else. Glycogen is especially important because there are some parts of the body that really, really prefer glucose. The brain is one of them. And so one of the major tasks that the body has is making sure that the brain has enough glucose to keep going well. Fatty acids are available through lipolysis, and if you haven't been eating for a while, muscles will use free fatty acids as one of their major fuels. They'll use sugar right after you've been eating, but if it's been long enough, they'll start using fatty acids as their energy, and that leaves more of the sugar available for the brain. I've mentioned already that protein should only be broken down for energy, at least, in the fasting state. That's why we call it breaking your fast in the morning. It takes about 12 hours to start using protein for energy. So you can just usually start detecting protein breakdown around the time that good, healthy people are eating their breakfast in the morning. And so it's a good idea to include a bit of protein as well as a bit of carbohydrate at that time. And the breakdown of protein turns out to be every bit as regulated as the synthesis of it, which should not have surprised anybody, but it did. And the two major routes for doing that are lysosomes, which are like a little stomach inside the cell, where there's a bunch of hydrolytic enzymes, and a neat little structure called a proteasome, which is kind of like these things where you can put trash in and get out stuff at the other end that can be reused for something else. So it's a little cylinder into which proteins that have been tagged with ubiquitin get stuffed into this cylinder and nibbled away at until it's broken down into peptides. Ubiquitin gets its name because it's one of the most highly conserved proteins known, and it's ubiquitous. It's in every cell it's been looked at. Initially, someone said, it can't be interesting, everything has it. So, yes, if everything has it, it probably is interesting. One of the important concepts you're going to want to have in mind is the idea of common intermediates. The idea that there are certain compounds that are shared along a pathway, they're shared between pathways, and that these provide a way for different pathways to be integrated. And these are some of the common intermediates that we're going to be seeing many, many, many times. So you might actually want to go home and learn these tonight. So if we start off with glucose, before we actually get off to carbon dioxide, we're going to stroll through three others. Glyceraldehyde, 3-phosphate. So it's an aldehyde, and it's a 3-phosphate. So the name is instructive. So here's a 3-carbon fragment. It then stands for several steps. It'll take the form of pyruvate for a while, still a 3-carbon fragment, but now with a carboxylic acid there, carbonyl carbon there, and a methyl group there. Pyruvate turns out to participate in a number of pathways, as does glyceraldehyde 3-phosphate. Pyruvate is one of the common precursors, not the only one, to make acetyl-CoA. And we're going to see that acetyl-CoA also is involved in a large number of reactions. So these different common intermediates turn up in amino acid metabolism. They turn up in photosynthesis. They turn up in the citric acid cycle. They turn up in fatty acid metabolism. So although we're going to meet them first in carbohydrate metabolism, we will meet them again and again and again. So they are good ones to know. You can also keep your eye out for them when we're talking about the different pathways as places where they intersect. Here's some of the ways that happens. So for instance, pyruvate can become an amino acid. And if there's an exchange of the nitrogen on that carbonyl oxygen, if it picks up a nitrogen somewhere, it's turned into alanine. And so this is one way of restoring amino acids, or to replenish the citric acid cycle. We're going to see why that would be important in just a little bit. You can also use pyruvate as a 3-carbon backbone to build something bigger. So if you can add a carbon dioxide to it, now that's not a favorable thing, so you're going to have to spend some ATP to do it. You can make a 4-carbon oxaloacetate, and we're going to see that compound in several places. This is an enzyme that's named fairly rationally pyruvate carboxylase. It takes pyruvate, it adds a carboxylic acid, CO2 to it. Now this is the, as I said, like a state view map quest. This is the overview view of what we're going to be talking about for the next several weeks. We're going to start by looking at glucose metabolism because it provides kind of a central location to look at everything else. If we feed glucose in, we're going to see that partway down we have glyceraldehyde 3-phosphate, some more processing to pyruvate to acetyl-CoA. But these then connect to other things. If you add in sources of carbon, you can end up making triacylglycerols out of the glycerides, glyceraldehyde. The pyruvate can interact with the amino acid pathways in a couple of different ways. Acetyl-CoA, again, can feed into the triacylglycerols or to being used just to give energy for the cell in general. So this is the sort of the high-level view of where we're going to be. Another recurrent, you're going to see it many times, feature of metabolism is oxidation and reduction. So if you weren't completely solid about that table we had back at the beginning of the most oxidized and the most reduced forms of carbon, go back and reread that and get that comfy in your head, because we're going to see a lot of oxidation and reduction. In general, catabolic reactions are often oxidative reactions. I mean, when you burn wood, you get energy out of it, right? So it's not too surprising that catabolism is often oxidative. To think of it in more chemical terms, oxidation is a loss of electrons. Electrons have a negative number, so if you gain electrons, you're going to become more negative. That's how you get reduced. If you lose electrons, you become more positive or oxidized. Electrons are never free by themselves. They're always on something. There's nothing like a copper wire in the cell that can take electrons alone. And so if you start off with some metabolic fuel that is relatively reduced, like fatty acids, the electrons from it will be carried through a cofactor to be used in other ways. And then the metabolic fuel is left in an oxidized state, and the cofactor is now in the reduced state. And here is an example of what that looks like. This is a cofactor we're going to see many, many times. This is nicotinamide adenine dinucleotide. So this is the nicotinamide ring up here, and that's why you need niacin, nicotinic acid in your diet, one of the B vitamins. Nicotinic acid is the precursor for this. It's on a ribose ring, just like nucleotides would be, and it's got this phosphodiester bond to an adenine. And so that's why it's nicotinamide adenine dinucleotide, or NAD, and we call it NAD all the time. NAD can come in this version, or it can have a phosphate down here on the adenine, in which case it's NADP. The phosphate has no effect on its energy. It's simply a tag that lets the cell know that this is a different category of it. We're going to use it for different purposes. So NADP has its reduction action up in the nicotinamide ring, just like NAD does, but it's used for different purposes, and we'll see why. So this would be in the oxidized state, I think. Is that right, Susan? And then be reduced from that. I'm not going to ask you to draw rings. Don't worry. And here it is in more detail so you can see it more clearly. Many times when we talk about enzyme cofactors, we can talk about the business end. That is, the cofactor as a whole may be this big, but the actual activity will be maybe 3 or 4 atoms at one side or the other. And I'll try to draw your attention to the business end of cofactors whenever that's useful. NAD and NADP are the most common electron carriers, so we're going to see them many times. And the two states you can find it in are in the oxidized state here with the plus, or in the reduced state, NADH or NADPH. So you can see it's picked up a proton and 2 electrons. The 2 electrons bring it from plus down to neutrality with that proton. The ring has gone from being aromatic to simply having a couple of double bonds in it also. So that's part of what makes it a useful carrier. One of the major ways in which those electrons are going to get moved around inside the cytoplasm. Sometimes you need to move electrons in lipids instead of in water. NAD and NADPH are both very water soluble, not very good for moving electrons in membranes. And for that, we have a lipid-soluble carrier called ubiquinone. This is one of those isoprenoid tails. You can see it has 10 copies of that puppy in there. So it's got a long isoprenoid tail. But the business end is the ring here. and it undergoes a change into these different states. It actually has 3 states that get seen at one time or another. Here's the fully reduced state with more hydrogen on it than it had before, more electrons. Here's the oxidized state, double bonds to the oxygen. So you can see that this carbon and that carbon are more oxidized than down here where they're just an alcohol. Okay? So if you start to recognize oxidized and reduced structures, it's easier to see which ones are which. Well, why would you use these electrons? Why do we bother having electron carriers anyway? And there are 2 major things that we can do with NAD or NADPH. If you have a reduced cofactor, and that reduced cofactor can be NADH or it could be ubiquinol, the electrons from that can go to 2 major things. We can either use it to build new molecules, or we can use it in oxidative phosphorylation, where the mitochondrion is going to use electron transport and generate available energy for the rest of the biological processes in the cell. And so we can use it for those 2 main purposes. I'm going to pause before we talk about the big picture because I want to make sure that Angie's new time is familiar to everybody, right? Because sometimes people come in late, and so if I announce it at the beginning, some people miss it. The new time for Angie's meetings will be 7 p.m. Wednesday, which would be today to start. And do you remember the room number?
Yeah, it's 433.
333 Bowen Science Building. And Angie pointed out that the Bowen building gets locked at 7, so try to turn up a little bit early. If you know you can't arrive by 7, let her know and we'll arrange some way to get you into the building despite all of that. Okay, any questions about that before we get back to metabolism? Okay. We're going to see this particular picture several times because this is the big picture. This is the overview into which most of metabolism fits. And so if you get it clear in your head, you're going to see recurrent patterns again and again, and it'll start to build up in your mind into one coherent thing. Some people find it helpful to actually make their own metabolic charts and to start, find a wall somewhere in your apartment, you can start putting stuff up. It really depends on how you learn best. The common features are that polymers are going to need to either be synthesized or degraded. And you can look at the degradation in the down arrows. So the polymers get turned into monomers. So proteins to amino acids, nucleic acids to nucleotides, polysaccharides to sugars, triacylglycerols to fatty acids. These monomers then, which are now a good deal smaller, can be broken down into yet smaller pieces, 2 and 3 carbon pieces. And part of what you're seeing is that these different kinds of monomers, the acids and sugars and nucleotides, converge on a fairly small set of 2 and 3 carbon intermediates. And these things then can be fed into the citric acid cycle. And the citric acid cycle, in turn, can feed electrons into oxidative phosphorylation, which will pump out ATP for the cell. The citric acid cycle can also provide intermediates for other purposes. So, for instance, it can provide 2 and 3 carbon intermediates that are going to be used for synthetic purposes. So go from the small up to the larger stuff and up larger again. In the down direction, it's going to be oxidative. The NAD plus is going to be turned into NADH. In the upward direction, we're going to be consuming reduced cofactors, NADP as well as NADH. So in these various reactions, you can be looking at, is the thing getting bigger or smaller? Are some of our shared intermediates here? There are a few places where inorganic things actually enter in. So carbon dioxide is released at the citric acid cycle. There are a few places where it's fixed to make new carbon intermediates. There are going to be a few reactions that involve electron transfers, and those are usually important reactions. And the biggest source of our ATP production will be through electron transport at oxidative phosphorylation. We'll see that there are a few other places ATP is made, but that's the big one. Any questions about the big picture before we move on to more of it? Okay. we're going to be talking mostly about kind of shared metabolism, the stuff that most organisms have. But there's quite a bit of variety. So for instance, a pine tree is metabolically more complex than we are. It has about 40% more genes than we do because it has to make everything. It has to make its own vitamins. It has to make its own amino acids and so on. So it has more pathways than we have. Dalmatians, like us, have forgotten how to make vitamin C. So they're one of the few mammals that need to have vitamin C. Most mammals can make their own vitamin C. And there are some where only a few organisms have it. So for instance, if you look at the stuff that poison ivy has that can give you awful welts, fortunately, there are only a few organisms in the world that can do that metabolic pathway. There are other metabolic pathways that are limited in distribution but are useful, like Taxol, which can be used as an anti-cancer drug. The various vitamins that we need are there because those pathways are no longer present in us. Here's just a couple of those. Thiamine is one of the B vitamins. We're going to see them in sort of some of the classes that they're useful for. We've met ascorbic acid very indirectly when we talked about collagen, because ascorbic acid is needed for collagen hydroxylation. Another big concept that we're going to come back to many times is free energy change. We don't usually try to measure the free energy of something. We usually look at the free energy change going from one state to another state. And to look at that with just a little bit of mathematics, when something is at equilibrium, a reaction that takes this form of A and B can go to C and D. At equilibrium, we can describe the tendency of that reaction to go in one direction or the other with something called the equilibrium constant. And the equilibrium constant is simply the products over the precursors as they would be at equilibrium. So this is one way. This number, K, the equilibrium constant, will tell you something about whether it tends to go towards C and D or if only a little bit of C and D is produced. That will be something that the equilibrium constant can tell you. Standard free energy change is the energy that is released when you start at standard conditions and go to equilibrium. And standard conditions are defined in biochemical terms. We have a delta G 0 prime, because this is a standard free energy that is the biochemical standard free energy. The prime means it's at pH 7. I understand chemists use pH 1 as their standard conditions. It's not very useful for biological systems. And so there's a standard free energy change that characterizes going from the standard conditions to equilibrium. And it can be related to the equilibrium constant through this equation. The T is usually going to be constant in biological systems. The R is a constant anyway. And so this is telling you basically that the equilibrium constant is related to the free energy change. Now, the actual free energy change, the energy that's available to actually do something in an actual living cell, is a little different. The actual free energy change will be the standard free energy change modified by how much you have of the reactants. If you've got a whole lot of the product, you're not going to be going in that direction very much. You're not going to have much of a free energy change. If you have a whole lot of the precursors, you may be beyond the point of the standard conditions. Maybe you get more free energy out of it. And so the actual free energy will be a function of both the standard free energy at biological conditions and the actual concentration of the substrates. So they will be related, but occasionally they'll flip signs. Occasionally it'll be something that has a positive delta G 0 prime and yet has a negative free energy change in the cell. And sometimes the other way around. So this is an important distinction. Any questions about this before we move on? Now, one of the reasons for thinking about free energy changes is that if you couple two sets of reactions, you look at the free energy change of the whole set. And what that means is if one reaction has a large negative free energy change, a large negative delta G, it can drive a reaction with a positive delta G that would normally not go. And so this is, as long as the sum of the two is negative, it will be a spontaneous reaction and go. And this is an explanation of why we keep finding ATP in so many places. Here's ATP drawn out again. You probably have some memory of this from nucleic acids. Here's the purine ring, here's the ribose ring, and here is the triphosphate. These phosphoanhydride bonds here are ones that are under considerable strain. Look at how those negative charges are all pushing together, so they're not terrifically happy together. Hydrolyzing either of those two bonds has a large negative delta G. And since they have a large negative delta G, they can drive reactions with a positive delta G. So both ATP and ADP have the potential to confer energy that way. And this is just one example of such a reaction. In fact, it's one of the first reactions that we're going to have. If we start with a glucose molecule that has nothing on it and inorganic phosphate and twiddle our toes around and wait forever, hoping to get glucose 6-phosphate, we would discover that the free energy change for that is almost 14 kilojoules per mole positive, meaning don't hold your breath. On the other hand, if we look at the hydrolysis of ATP to ADP, that has a large negative delta G. Since these things add up, that is, we can talk about glucose plus ATP going to glucose 6-phosphate plus ADP, conserves all the pieces, right? The reaction overall is going to have a negative 16 or so kilojoules per mole. It will proceed in a spontaneous way. And so we see that ATP is able to act as a messenger boy between reactions. If we look at catabolic reactions, breaking things down, some of that energy can be captured in ATP. If we look at anabolic reactions, building them up, we can use that energy in the production of it. I have said this, but I obviously wanted to say it twice because I made a special slide for it, that the concentration of the reactants makes the difference, that the actual delta G will depend on the actual concentrations. Something to keep in mind is that some reactions have reactants or substrates that aren't soluble enough to get the concentrations high enough to do that. So it's there in principle, but it wouldn't necessarily be in practice in every case. Now, what's so magical about ATP? Well, there's several things. I've already pointed out the charge repulsion, that there's all this negative charge kind of pushing itself apart. So that's one thing that tends to favor the products rather than the ATP. There's resonance stabilization. So you can see that you can draw the structural groups around the phosphate in a highly resonant form when it's inorganic phosphate. It's more constrained in the phosphoanhydride bond. And so this is a more resonant product. So resonance stabilization favors the breakdown products over the ATP itself. Something that's very important to remember about ATP, and part of the reason why it's such a magical molecule, is that it is kinetically stable, but not thermodynamically stable. That is, it has a large free energy change for hydrolysis, but there's a big barrier between. So the rate at which it spontaneously breaks down is not very fast. The free energy change favors the breakdown, but there's a big barrier in between, so it doesn't happen very fast. And that means that the ATP lasts long enough for enzymes to use it for various processes. So it's, kinetically speaking, ATP is stable, meaning you can keep it in the test tube and it'll be there for your experiment the next time. Thermodynamically, it wants to break down. And that combination is what makes it so useful as the energy currency in the cell. If it had a small delta G change, not much energy to get out of it. If it weren't kinetically stable, it wouldn't be there long enough to use it. So it's this magical combination of both at once that really makes the nucleotide triphosphate so amazing. Now, there are other things that can carry energy, and some of them are almost as important. Reduced cofactors can be thought of as carrying energy. They're carrying electrons, and the transfer of the electrons has associated free energy change. So both NAD and ubiquinone in their reduced forms can be thought of that way. For short-term use, phosphocreatine is a compound that can transfer roughly equivalent energy bonds. So here is phosphocreatine. Here's the structure of it. You don't need to memorize it. And that phosphate, which is on a nitrogen, which is kind of funny, isn't it, has a high enough energy that you can use phosphocreatine to make ATP. All the nucleotide triphosphates, CTP, GTP, UTP, all the nucleotide triphosphates have the exact same free energy change associated with them. Because the energy is in that triphosphate tail, it's not in the base. All that the base does is determine which enzyme is going to use it. In theory, we could have had all the energy currency running off of UTP, but we don't. We use ATP. But because these things are all in equivalent energetically, enzyme nucleoside diphosphate kinase can move those phosphate groups between them pretty much equivalently. They just kind of flow downhill whichever ways it's needed. So if you have ADP and CTP, CTP can give its phosphate to ADP, and now you've got ATP and CDP, because those phosphates are energetically equivalent. The absolute bare bones approach to phosphate bonds is in microorganisms where they actually store phosphate as polyphosphate. Now, most microorganisms are short on phosphate. That's a rare element in the environment, so it's not surprising that they store it. But they store it in this polymer of phosphate, which has a phosphoanhydride bond between each phosphorus. So they both store the phosphorus and they store energy. Each of those phosphoruses is potentially capable of transferring a high energy bond to something. This is kind of an interesting example of how all of this adds together in our practical terms. That we see the contributions of different energy systems at different times and in different contributions. So ATP is available very quickly in a matter of seconds. And we can see ATP fueling things like, shot puts and high jumps, things that happen in a flash. Those are going to be driving pretty much pure off of the ATP that the cell has at the moment. If you've got a little more time, like up to 10 seconds, you can draw some energy out of your phosphocreatine and recharge your ATP. Typical things like that would be sprints or the football line about to crash into somebody. Both of these are happening anaerobically. There's no oxygen needed for them to do something. So these are things that you've got sort of ready to go like a battery. You can use it right now. You don't need any oxygen. But you can use it up. Part of the point of the short bars is to show you that there isn't much of whatever that is. Now you can, if you've got a bit more time, you can start to regenerate your ATP through glycolysis. Glycolysis is itself an anaerobic process. And if you do that, you can accumulate an oxygen debt. People talk about lactic acid and so forth. But for short distances, glycolysis is going to be the thing that's going to replenish your ATP. We can't keep that up for very long. If you want to do anything for much more than a minute, minute and a half, you start to get into the aerobic processes, and then you need to get oxygen in, and you're going to draw in additional metabolic pathways. We'll talk about those as we get to them. Oxidative phosphorylation in the mitochondria, and that gets used for the rest of the way. Some of the neat things here, over here, sort of little places we see this turning up. White meat is anaerobic meat. The reason that chickens have white meat in their breasts is because chickens can't really fly anywhere. If you look at wildfowl, they'll have dark meat there because they fly for miles at a time. They do it aerobically. Whereas chickens stand on their legs a lot, so they do that aerobically, and they have mitochondria there that make it red. Ultramarathoners are using oxidative phosphorylation certainly to do that, but they're also driving it off of fat metabolism. The fat is the fuel that drives that especially. And the crocodiles are in here because of their odd approach to all this. So if you have ever seen these nature movies, the crocodile like, I make a good crocodile. That is an anaerobic thing. That's fueled off of the ATP and the phosphocreatine. And they can do that for actually more than just a few seconds, but not for very long. And they build up a ferocious oxygen debt. So the crocodiles then have to lie limp as a log for quite a while to pay off their oxygen debt. They have to sort of decide, am I going to go for this one or that one? Because after they've lunged, they're going to have to go back and pay off the oxygen debt. Another important high-energy compound takes advantage of a different kind of bond. These are the thioesters. CoA is the shorthand term for this. Coenzyme A is sometimes the other name for it. And it forms compounds. Here is the schematic of an acetyl-CoA. This bond between the acetyl group and the sulfur also has a free energy of hydrolysis fairly close to that of ATP. We're not completely sure why. I love that there's still things in biochemistry that aren't completely understood. And the place where this is all happening, the business end, if you like, is this sulfur group here. And there's a pantothenic acid residue sort of holding on like a swinging arm. So that's another one of your B vitamins. And it's on an, oh, look here. Here's an adenine ring, and here's the ribose. This is an ADP molecule. So coenzyme A is another thing that uses an adenosine ring as part of, or an adenine nucleotide as part of its handle, if you like. CoA is both used synthetically, so it's part of anabolism, and it's used to make energy for catabolism. And that's the end of the slideshow. I do really commend to you that animation of the cell. I am desolate that they don't have flash here. Please take a moment to look at the animation on the web.
In an NMR facility, and their clock is about 30 minutes off. So it says 3:15. I'm like, okay, I have 15 minutes to get there. And I'm walking, and I don't see any students out there, and I look at the clock, I'm like, oh,